This topic will cover scientific method and how you relate observation and experimental analysis in your field of study. Why is this useful? Let's just say some of you become nurses, some of you become a radiologist, and so on, or just enter any healthcare field. Maybe, say, 10 years down the lane, you feel you want to do something else. Okay, and how can you use your field of study otherwise? This is where clinical research comes in. Okay, so clinical research involves different stages of research in clinical studies. Let's just say, for example, COVID-19 vaccine, uh, you have preclinical studies, which is done on lab animals in the laboratory, and then you have clinical studies. Okay, clinical studies is done on actual patients in different stages in the hospitals. And as clinical researcher, you need to have the fundamental knowledge of the scientific method. Not only that, but you need to know the science behind it. Okay, so let's just say you are a nurse working in the cardiovascular field for the past 10 years. You can use that skill to enter clinical research in uh, uh, medicines for cardiovascular diseases, right? Or you work in the oncology department as a clinical researcher, you can enter a clinical research related to oncology, right? So that's why the study of clinical method is important. One of the things you'll be doing is study anatomy and physiology, obviously, and you will do this by observing the patient's recording data. And sometimes when you write report, you will ask a question regarding the scientific process, depending on your job responsibility and depending on the requirement. Okay, so how can you relate the observation and experiment to your field of study? So let's ask a question. When you were a kid, you were told music made plants grow. So based on your general observation, you form a hypothesis that music makes the plants grow. But you won't know for sure unless you have an actual experiment. Okay, this is where scientific method comes in. Now, in a scientific method, first you make an observation. What's going on over here? Yeah, so first you make an observation, then you ask a question. Okay, your observation is that music might make a plant grow. So you ask a question, does music make plant grow? And then you form a hypothesis that music makes plant grow if it is kept near a window. Okay? And then you conduct an experiment. Your experiment will have several different things. One is the control. Okay? So control is something that you know the outcome for. Okay? So you have a plant that grows normally without music. Okay? So no matter which plant you put in sun, you've seen it growing, right? So you know a plant is going to grow in sun. You're going to keep that by the window. And then you'll have another plant with music by the window in light, just like the first plant. The only difference would be you'll have uh, a plant plus music. Okay, I'm going to write this down. Okay, so your control will be plant in light, okay, because you know the outcome. And then your variable one will be the plant plus light plus music. Then your variable 2 will be plant let's do minus light plus music. Then variable 3 will be plant minus light minus music. Okay, and then finally you realize that there is no difference between your control and the plant kept in light with music. Okay, and then when you keep the plant in the dark, it's no difference over here. When I keep the plant in the dark, because there is no light over here, both with and without the presence of music, the plant does not grow. Okay, so you come to your conclusion that your hypothesis is not supported. So what was your hypothesis? Is that plant grows in the presence of music in light. Okay, so you come to the conclusion that music is not essential for growth of plant. Okay, so on the basis of your results, you can accept your hypothesis, or you can say that your results do not support your hypothesis. So you reject your hypothesis. Okay, I'm not going to ask you too much about scientific method, but just you need to understand that in any experiment, you have a control that you know the outcome of, and then you have variable with different variation in parameters to observe the changes in experiment. So in an experimental design, you have the sample size. Sample size is the number of your samples. If you are doing a test on 100 patients, 100 is going to be the sample size. Then an experiment will have controls and variables. Okay. Control is something that is untreated for comparison. So you generally know the outcome of the control. Okay. And then you have an a dependent variable, something that you cannot control. Let's just say that the amount of glucose affects heart rate. That's your experiment. Okay. You cannot control heart rate. So that is going to be your dependent variable, something that you cannot physically control. And then you have independent variable. Independent variable is something that you can control. So you give a patient 5 milligram of glucose, another patient 10 milligram of glucose, another patient 20 milligram of glucose. So this is something you can control. So glucose becomes your independent variable because you can control the amount of glucose. Okay. Now in clinical research, there's something called a psychosomatic effect. Psychosomatic effect is something that takes into account the subject's state of mind on the physiology. And this is controlled, or this is tested by giving one group a placebo. Placebo is a sugar pill, basically fake treatment to the control group. They don't know that they're getting a placebo, okay? But in a clinical trial, there's always a placebo group which acts as a control. So the group thinks they're taking medicines, but they really aren't. Okay, and sometimes a placebo itself makes a patient feel better. Okay, these are called psychosomatic effects. Okay, now in any experimental design, it is important that there's no experimenter bias. So it is avoided with a double-blind study, meaning both sides, both parties do not know what condition they are testing on a patient. Okay, and finally, it involves statistical testing and data observation, which is followed by peer review by a group of scientific experts in that field. Okay, so this is all you need to know about scientific process. It might help you in the future.